everybody. Today we're going to talk about the background you need to be able to do the biochemistry lab that we've got planned for this week. So in this biochemistry lab, it's probably one of my favorites actually, we are going to look at a bunch of different food items, both food for us but also food for bacteria, because we're going to work with bacteria a lot this semester. And we're going to look at those food items and figure out whether they have carbohydrates, proteins, or lipids. These are 3 different types of macromolecules. There are actually 4 different types of macromolecules, but we're not going to work with nucleic acids this week. We're going to hang on to that for a little bit later. So our goal is to determine whether these foods have any of these carbohydrates, proteins, lipids, or nucleic acids. So to determine that, we need to know what is in each of these macromolecules, what their monomer and polymer forms are, what part of those macromolecules are involved in the biochemical tests we're going to do, and then which biochemical tests or which biochemical reactions will help us determine these compounds. All right, so let's start with carbohydrates. So carbohydrates are made up of monosaccharides. Monosaccharides include things like glucose, but also other single sugars like fructose or galactose. Monosaccharides don't count things that are made of multiple monosaccharides, so multiple monomer parts. So things like lactose, the sugar found in milk, is going to be a combination of glucose plus galactose. Sucrose, the sugar that you might stir into your coffee or tea or use for baking, what we would call table sugar, that is actually a disaccharide made up of glucose and fructose. And then maltose, the sugar that is in maple syrup and has that particular maple syrupy smell, is a disaccharide made up of 2 glucose molecules. So the thing that we're going to be testing for in our monosaccharide tests or our monosaccharide experiment is specifically these single ones where you have single monosaccharides by themselves. All right, in these, there are lots of free OH groups or lots of free hydroxyl groups, and these free OH groups or hydroxyl groups are very reactive in the particular biochemical test that we're going to do here, which is Benedict's test. All right, so as you complete this test, you'll add the Benedict's reagent, you'll heat the sample, and then if there are monosaccharides present, you'll get a result from that. I split this carbohydrates up because we're actually able to test for 2 different types of carbohydrates. We can test for the monosaccharides with Benedict's test, but we can also test for the polymer form of carbohydrates, in this case, starch. So there are a couple other types of polymers as well, things like cellulose and glycogen, but we're specifically going to be testing for starch. And this starch test, it looks for a particular 3-dimensional structure that happens when you have a bunch of starch molecules. It turns out these starch molecules bend and twist back on each other and themselves, and they form these sort of looped structures. So it's these looped structures or these spirals that is able to react with the Lugol's test in order to indicate that starch is present. So the second macromolecule that we're going to test for are going to be proteins. And proteins are made up of the monomers amino acids. And amino acids are attached together by peptide bonds to form polymers of proteins or polypeptides. All right, so the part of the proteins that is reactive in the biochemical test that we're going to use is actually that peptide bond. That peptide bond is able to react in the Bioret's test, and it will wind up causing a color change, indicating that there are proteins present in that sample. So for the protein test, what you're going to do is you're going to take each of your samples, you'll denature those proteins by unfolding all that tertiary and secondary and quaternary structure, leaving you just with the primary structure so that all those peptide bonds are open and exposed. Then you're going to add the Bioret's reagent, and if there are proteins present, then you'll get a color change at the surface where that Bioret's reagent is added in order to indicate the presence of proteins in that sample. And then the third and final test that we're going to do is looking at lipids. And the particular test we're going to run here is called Sudan's test. And Sudan's test works on lipids because lipids are hydrophobic. So that's the feature that unifies all of the lipids together, is basically that they're made of long chains of hydrocarbons. And as a result, they're hydrophobic because they don't have anything that can make hydrogen bonds. As a result, all lipids will be hydrophobic and therefore will be able to bind to and interact with hydrophobic things like a hydrophobic dye. And that's exactly how we're going to do Sudan's test. We're going to take those lipids and we're going to stain them red, showing that they're hydrophobic. Lipids, unlike carbohydrates and proteins, don't have really straightforward monomer and polymer setups. They're not made of something really simple, like a really simple monomer that gets put together in long chains to make a polymer. They're a little bit more complicated. So the building block structure for lipids is a glycerol plus the fatty acids. And depending on what type of larger lipid you're making, it might be a glycerol plus 3 fatty acids, or so that would be if you're making something like just a fat or an oil. Or it might be a glycerol plus 2 fatty acids plus a phosphate head group. And that phosphate head group would allow you to be making a phospholipid. So the polymers are a little different than what you might see for carbohydrates and proteins because they're not long strands of molecules. The last set of macromolecules are ones we're not actually going to be testing for. These are nucleic acids. So they're made up of nucleotides, and these are used to make long strands of molecules, things like deoxyribonucleic acid or ribonucleic acid, so DNA or RNA. These nucleotides are present in all cells, so any food item made from cells will have nucleotides present in it, but we're actually not going to be testing either of those things today. So I'm just going to fill in that these are not applicable. We're not going to be looking at that. All right, as we go on and do these tests and as we do these experiments to determine the different macromolecules present in each of our food items, one thing that we need to think a lot about are controls. And controls are components that are designed into the experiment to help you determine whether or not the experiment is working as you intend. So every experiment has to have controls associated with it, and it's important that you have both negative controls and positive controls. So the negative control is designed in such a way that you know that it won't have a response. So basically, if you're testing something to see if it has glucose present, you need to have a negative control that you know does not have glucose so that as your test, you know what that negative result looks like. It's a way of checking and making sure that the experiment is working the way you intend and that all of the different parts and all of the different features of the experiment are working accurately and working correctly. So you need to have a negative control, one that you know will not have a response, and you need to have a positive control, which is the opposite. It's one where you know that you will have a response. You will have an appropriate response. So let's go back to that glucose, for example. So let's imagine that you wanted to test and see if you had a monosaccharide or glucose present in your experiment. So you would have 3 things that you would test at the very least. One would be your negative control, something that you know for certain does not have glucose. Water is a really great example. Water is just water. So pretty straightforward there. And then you'd have a positive control, something that you would know would have glucose. So maybe you would want to do something like glucose itself. You could test straightforwardly, negative control, water, no glucose is present. Positive control, one where I know glucose will be present, and I'll test glucose. Then you could have your experimental groups. And your experimental groups is anything unknown. Anything where you don't know what the result will be. So in your pre-lab for today, it asks you to think about how you might test different soda samples. and see whether they are made with real sugar or with fake sugar. So do you have real 7-Up or do you have Diet 7-Up? Because Diet 7-Up would be made with artificial sugar. Those could be your experimental groups where you're using water and glucose as your negative and positive control. So that's one thing to think about as you design and think about these experiments. All right, I'm looking forward to this fun lab this week, and I'll see you all soon. Have a great day. Bye.